And I'm very honored to be speaking today as a result of this kind invitation from the World Health Organization and the organizers on plasma fractionation, a historical and contemporary view. This is a subject which is extremely close to my heart and to which I have devoted my whole career. The aims and objectives of this session will be to assess the historical development of plasma fractionation. We will review the basic technologies, and then I will analyze some of the so-called product drivers of the sector, specifically albumin, factor VIII in the immunoglobulin, and then I will summarize and come to some conclusions. So, who developed plasma fractionation? Well, the very first fractions from plasma were actually manufactured not from human, but from animal plasma in the form of crude preparations of antibodies, which were in those days called antitoxins for various infections. And this was done more than 100 years ago by the famous German immunologist Emil Behring. However, our field really started with the work of this gentleman, Professor Edwin Cohn, from the United States, who was a professor of biochemistry in Harvard University. And in the 1920s and 1930s, Professor Cohn and his team developed ways to purify most of the proteins which are found in plasma. And many of the methods which he used are still applicable today. But the important thing to note is that as a result of his work, he got the attention of the United States military. And as World War II broke out and America got involved, he was directed, he was commissioned to develop a preparation of human albumin as a stable blood substitute for people who are injured. And he did this using the scheme which you can see on your slide, on this slide, whereby he developed the system where proteins were sequentially precipitated from plasma using several variables, including the use of cold ethanol, which gradually brought the protein mixture to a state that the final precipitate was in the form of albumin. And this was, of course, his charge. This is what he was asked to do. We will come to some more aspects in a few minutes. The technology for plasma fractionation has evolved significantly. In the slide on my left-hand side, you can see the very first plasma fractionation plant, which Cohn and his co-workers used to develop the system during the Second World War in Harvard University. A fairly primitive plant, as you can see there, capable of fractionating relatively modest volumes of plasma, whereas on the other half of the slide, you can see a modern Cohn fractionation plant utilizing pretty much the same kind of equipment in the form of tanks in which the plasma and the reagents are added, but in a much more sophisticated fashion using a high level of automated control in order to adjust the pH, add the ethanol, and do all the other steps. As I was intimating, the very important aspect of this work was that it had to be useful in terms of supporting people who were injured during battle, including people who were severely burned. And on the slide, you can see the very important observations on some of the very first injured people who were given albumin purified by Cohn fractionation. And these were people who were injured, sailors who were injured after the attack on the United States fleet in Pearl Harbor. And you can see from this slide that many of the injured people were able to be resuscitated and very much made an improvement and survived by utilizing the albumin preparation. So this was a very important illustration of the benefits of albumin. So I have called Kohn the first pioneer because he developed a system, a technology whereby we could purify plasma proteins. And as I said, the very first protein was albumin. The second pioneer is this gentleman here. Professor Kohn is no longer with us. He died in the early 1950s. But this gentleman, thankfully, is, and he is my good friend, Dr. Curling, who in the 1970s developed another technology for purifying proteins from large volumes of plasma, and this was through the use of various types of chromatography. And chromatography, as you know, is a very different technology from precipitation using ethanol. In chromatography, we have these large columns, which you can see on the other half of the slide, which are packed with specific gels, we call them, which allow the separation of mixtures of proteins, such as are found in plasma, using several biochemical principles based on charge, based on size, and so on. And so we have these two main technologies. And you can see, for example, here, that in terms of one of the most important preparations from plasma, we can utilize also chromatography, as you can see on the right-hand side, in contrast to the utilization of ethanol precipitation developed by Kohn. On the left-hand side, this is showing actually a product purification which was utilized, but which is now no longer utilized by the company involved. But it serves to show the principle that proteins can actually be purified with both Kohn fractionation and with chromatography. And in fact, what we have today in most plasma fractionation plants is a combination of the two technologies. Both Kohn fractionation and chromatography are utilized in the same purification schemes by many companies which manufacture plasma proteins. Let me now say something about the industry. When this industry started with the purification of albumin by Kohn, it was a very small industry, and it stayed that way for many years. You can see, however, that in the past 20 or so years, there's been a very significant increase in the growth of this industry. So you can see that over the first 20 years or so of the 21st century, this industry has grown by about 8% every year. And it now has a considerable market value of more than $20 billion. And this growth continues even in the years after this slide was constructed. This is because we have now discovered that there is a very wide range of plasma proteins which can be used clinically. And you can see these on this slide. I expect that the organizers will make the slides available to anybody who wants them, who can return then to scrutinize the number of therapeutic proteins which are available to be purified from plasma. It's important to note that the plasma fractionation process has its own economics based upon the fact that it is a cracking process, a little bit similar to the generation of several products from the crude material, which is oil. So that if we look at any fractionation company and we look at the so-called concept of so-called plasma fractionation economics, we look at the way in which companies fractionate all their plasma. And every single liter of plasma generated by the suppliers to the companies is nowadays extracted for immunoglobulin. Most of the first few thousand liters of plasma also have the albumin extracted. And then, depending on the company's capacity for technology, other proteins are removed until, as they approach the last liter, which they fractionate, it will generally involve only fractionating the immunoglobulin. As I shall discuss in a few minutes, immunoglobulin today is the most important protein of the plasma fractionation industry. A word about the raw material plasma. There will be other talks in this series which will address this in more detail, but I will just very quickly summarize that we have two main types of plasma, which are generated using different technologies. First of all, and most relevant to countries which still have emerging economies, is recovered plasma, which is extracted from whole blood donations as a sort of byproduct. We call this recovered plasma. It is an entirely valid and good quality material to be sent for fractionation. And then there is source plasma, which is extracted from donors using the process of plasmapheresis, whereby the red cells are returned to the donor, the plasma is generated on its own, and this allows much more frequent and higher volumes of plasma to be collected than with recovered plasma procurement. And you can see on this slide how nowadays in the overall global plasma economy, it is in fact the source plasma from plasmapheresis, which is the dominant material which is used for plasma fractionation. However, it is extremely important to note that we still have a significant volume of recovered plasma. This is extremely critical in the supply of plasma derivatives, especially for countries who do not have access to plasmapheresis and sophisticated fractionation. And it is also important to note that large volumes of plasma, which are potentially useful for fractionation, are nowadays not used because the recovered plasma collection does not come up to standards. Standards for this kind of activity have been developed by the WHO, and you will see them in some of the literature which is noted to you at the end of this presentation. I want to now go into the actual products, and I will just remind you that since the inception of industrial plasma fractionation during the Second World War, a succession of protein therapies isolated from plasma have determined the volume of plasma which requires to be collected and have also shaped the economies of the industry. These so-called plasma drivers have successively included albumin, coagulation factor 8, and for the past 30 years, as I have intimated before, immunoglobulin, which is nowadays available in both an intravenously administered and a subcutaneously administered form. On this slide, you will also see a publication by myself and some other collaborators in Italy regarding the importance of self-sufficiency, particularly in immunoglobulin. This is a topic which will be presented to you by other speakers in this series. So let us talk now about the proteins in terms of their chronological appearance on the clinical landscape as I have described. And the first of these drivers is albumin. And I have already said that albumin was initially developed as a product to address severe injury and blood loss. And this function of albumin is so-called oncotic in terms of the physiology of the albumin molecule. You can see the albumin molecule pictured here in terms of its primary and also up to its tertiary structure, and the oncotic value of albumin, the capacity of albumin to maintain the volume of blood and plasma, is related to those aspects which you can see on the left-hand side of this slide. And for many years, this was indeed the main usage for albumin. However, our knowledge of albumin in terms of biochemistry and physicochemical characteristics has grown enormously. And you can see on the right-hand half of the albumin molecule, you can see that there are a number of other functions which we classify as non-oncotic, and these functions are extremely important in the physiology of albumin and are of also significance in, as I will show, in some other diseases, including sepsis and liver disease, in terms of maintaining physiological capacities such as the antioxidant capacity of plasma, immunomodulation, and the stability of the endothelium itself, which is extremely important to ensure avoidance of sepsis and other kinds of shock. Here is an important series of studies whereby we have seen investigators studying the survival of patients who have severe sepsis who have been given albumin. And these are very large-scale clinical studies. They are all available in the literature if you look them up. And you can see that in every study, we can see that albumin does develop a benefit for people given the albumin compared to crystalloids, compared to salt solutions and similar products. However, in all of these large studies, we have not seen a significantly enhanced effect. We have seen an enhanced effect, but it did not reach clinical significance. However, on the study which you see furthermost on the right, the so-called Albios study performed in Italy, where I am speaking from at the moment, a subset of patients who were suffering from severe septic shock actually showed a significant benefit in survival when given albumin. So this important observation still requires further confirmation. However, it does indicate a potential benefit of albumin in this extremely important group of patients who are severely ill. A very interesting and important other use of albumin, which has emerged in the past 15 years or so, is in patients with cirrhosis and different types of liver disease. And recently, we have had a very important study in Italy as well, the so-called ANSWER study, in which albumin was administered on a long term to patients with decompensated cirrhosis. And this study demonstrated very significantly, without any equivocal in these results, that there was a survival benefit in the patients. It has to be said that there is some controversy because subsequently some studies which attempted to address similar patient populations could not confirm this extremely large study, which was done in Italy. However, we have to address the fact that whereas albumin is able to be substituted with salt solutions for the indication for which it was developed, which was injury and shock, albumin, in terms of these pharmacological effects, which are contributed by the more sophisticated structure of the molecule, cannot be substituted with any artificial drug. So our attempts to exploit the wonderful chemistry of albumin continue. I want to make the point that albumin from different manufacturers can actually be different in terms of its properties relative to these issues which I have described to you in terms of the pharmacological effects. And we can see this in these studies, which are shown on this slide, whereby different albumin preparations are showing differences in terms of their capacity to bind drugs and to protect the endothelium from inflammation, which is what these markers are measuring. The references are there for you to study if you are interested in this. The next driver, which came about over the history of fractionation, was factor VIII. I think everybody who is listening to this talk knows about the famous disease hemophilia, which was made famous because it was in the British royal family through Queen Victoria. And since Queen Victoria's children married many of the monarchs of Europe, this also found itself in several of the European royal families, and most importantly and famously in the last royal family in Russia, in the form of hemophilia to the Tsarevich, the son of the last Tsar, who was unfortunately born to his mother, a granddaughter of hemophilia, a granddaughter of Queen Victoria, who was a carrier for hemophilia, who gave the disease to her son. Hemophilia is a disease on which there is a massive literature and is mostly characterized by excessive bleeding because of lack of the coagulation factor VIII, and this results in bleeding in the joints, which you can see in this other half of the slide, and crippling the patients who are unfortunate enough to suffer from this disease. As I did with albumin, I mentioned the pioneers in relation to developing treatments of hemophilia. One of the most important products in the early years, in the 1960s, was cryoprecipitate, which was actually developed not in the 1960s, but in the 1950s by the French physician Émile Rémigy, who also did a thesis on the way in which he could purify cryoprecipitate from frozen plasma, allowing it to be concentrated in factor VIII and used to treat hemophilia. Since this treatise was published in a little-known French journal in French, it was not much noticed, and 10 years after its publication, the American biochemist Dr. Judith Poole rediscovered this without knowing of Rémigy's work and published herself a method for making cryoprecipitate using closed plastic bag systems in a blood bank, and this totally revolutionized the treatment of hemophilia. Of great importance as well were the group of people which you can see furthermost on the right-hand side of the slide, the Swedish investigators, the couple Blombach, Margarita and Berger, as well as the very famous clinician Ingemar Nielsen, who treated patients with the very first concentrates developed in Sweden, which were not cryoprecipitate, and which allowed Sweden to be foremost in the treatment of hemophilia from the very early stage. This shows us the evolution of hemophilia therapies, and as you can see, and as I have intimated before, over the 1960s and 70s, we had the factor VIII concentrates in the form of cryoprecipitate and also intermediate and high purity concentrates developed from plasma. So plasma is obviously a very important source, and fractionation of plasma to factor VIII concentrate is very important. In the established economies, this has been mostly replaced by recombinant factors and other types of biologics, which I shall mention very soon. But it is very important to note that as a result of developments, we now have very safe plasma-derived factor VIII concentrates, which are extremely important for countries with emerging economies, where the ability to access expensive biological drugs is not as high as in the Western countries. A very important development in the last 8 years or so has been the gradual development of gene therapy as a possible cure for hemophilia. This has not yet fully come about, especially for hemophilia resulting from factor VIII deficiency, but we all are in hopes that this will happen. In this slide, I am showing you the remarkable progression and developments in the number of therapies which are available for people with hemophilia around the world, and in particular in people in established economies. And you can see that the number of these concentrates has increased significantly over the first 20 years of this century. But you can see that, very importantly, we still have in colored in green on the slide, the first concentrates are all plasma-derived concentrates. These are still very important. We also have recombinant concentrates in the ones which you can see in red, and I have especially put in large letters the product called Hemlibra, which is a biological product. It is not a factor concentrate. It works in different ways in influencing the coagulation mechanism, but has acquired a high level of popularity in use in particularly the United States. This is not a subject which I will enter into further because it is beyond the scope of our talk on fractionation. You can see in this slide how important this availability of these products has been. You can see that the life expectancy of people with hemophilia was, especially in the severe form, very low when you compare it to the normal population up to the late 1950s, and then it started to go up significantly, very high, much higher over the 1960s, 70s, and 80s. We unfortunately had a blip in this exciting development in the late 1980s to the 1990s because of the development of infections coming from the plasma products which entered the blood supply during this time in the form of AIDS and hepatitis C. Fortunately, the industry was able to develop concentrates which nowadays are 100% safe from these infections, and the progress towards enhanced survival continued to our days, so that in countries which are able to access the product, we have a full life expectancy for people with hemophilia. And you can see this in this slide, where you can see the prevalence of hemophilia A plotted against factor VIII usage, so that when we have about factor VIII usage of 5 international units per capita in the population, the prevalence stabilizes to about 6 people per 100,000 inhabitants. This shows that the people with hemophilia are living long enough for them to be detected in the population, and shows the benefit of having access to factor VIII at these levels. At lower levels, we have a less high quality picture, but we still are able to save a lot of lives and to enhance the quality of life of people with hemophilia in the ways which you can see in the scale at the bottom of this slide. I emphasize the continued importance of plasma-derived factor VIII, not just because it is a more significant product for countries which are unable to access expensive biological drugs, but because it is also a product which an important study performed worldwide some years ago, the CIPID study, which showed that this product is much lower in risk in terms of developing inhibitors to factor VIII, which is nowadays the most important complication from treatment with products for hemophilia. Comparing two populations of patients, one with given plasma-derived and one given recombinant factor VIII, there was a significantly lower incidence of inhibitors in the patients given plasma-derived factor VIII. This has implications for the clinical use of concentrates for hemophilia, which I will not get into detail in this talk, but I emphasize the continued relevance of plasma-derived concentrates. I now come to the last product which I will discuss in detail, which is the current driver, which is immunoglobulin. And immunoglobulin, ever since it was developed, has been an increasingly important product. Recognizing again the pioneer in this treatment, this is Colonel Ogden Bruton, who was the first person to treat a child with immune deficiency, which led to severe infections, using a preparation of purified immunoglobulin generated by the fractionation system as you can see, nearly 70 years ago. Since then, the preparations have continued to be developed. You can see how in the group of patients characterized by the very first patient who had X-linked agammaglobulinemia, the ability to have a preparation to treat the disease was enhanced over several decades and resulted in a much enhanced survival of these patients. So you can see that a cohort which was assessed in 1955, unfortunately the survival in terms of years of life was quite low, whereby we go to the situation 50 years later in 2006, where Ig therapies, immunoglobulin therapies, were widely available, you can see that the survival is significantly higher and approaches that of normal individuals. And this is the same also in the other very important group of immunodeficiency, which is common variable immune deficiency. Again, we have several cohorts here studied in the United Kingdom. And the message from this slide is that if you look at the cohort studied in 2003, the survival of the individuals affected with common variable immune deficiency is much better than those studied in earlier years in the 1970s. As I have said before, immunoglobulin is the most important product in terms of the industry today, in terms of both the volume of plasma needed to purify because of the volumes of product needed, but also in terms of the economics of the industry. Excuse me. Immunoglobulin, as you can see, as a percent of worldwide plasma product sales, now represents about half the value of the industry. And this has grown significantly in the past 20 years and is relatively higher than for other proteins. So this, in terms of the economic viability of the plasma industry, is a very important product. And you can see that the demand continues to grow. This is immunoglobulin consumption as shown in 2018 and thus projected towards the later years of our current decade of the 2020s. You can see that North America, as always in any matter involving medicine, is the most important user of immunoglobulin, but it is widely used and the amount is projected to continue to grow across the whole world. And we need more and more of this product in the countries where the economies are still emerging. Importantly to note is what is immunoglobulin used for? It is, of course, used for, as I have indicated, in the similar way that hemophilia as factor VIII is used to treat a deficiency of the protein. Immunoglobulin is also used to treat a congenital deficiency of immunoglobulin G in the form of primary immune deficiencies, which I have described. However, nowadays, one of the most important groups of indications for immunoglobulin, and the single largest group, is actually the neurological autoimmune diseases, such as Guillain-Barré syndrome and chronic inflammatory demyelinating polyneuropathy, multifocal motor neuropathy, which are immensely important in terms of consuming the very largest section of the product available on the market in the Western economies. In the still emerging economies, this is not the case, and the product is mostly used, very importantly, to save the lives of people, such as I have described before, who are deficient in the product at birth. It is very important to understand that we utilize evidence-based medicine in plasma derivatives, as in everything else. And it's important to note that for the primary immune deficiency, there is absolutely no doubt that the evidence is that there is no other product which can treat well these patients who are deficient from birth in IgG. With other diseases, there are other products which are perhaps able, if not to the same extent, to address in terms of the disease and the symptomology. And one of them is the disease chronic immune demyelinating polyneuropathy, which, as I've said, is very important, but which, as you can see from this review, from the Cochrane collaboration, can sometimes be also treated with other types of medication. It is important to understand these things in terms of our ability to prioritize the very scarce resource, which is plasma and immunoglobulin, in terms of patients receiving it. Just to round off my talk, I will just mention some other products which are important, not as important as the other products in terms of numbers of patients, but which are certainly important for the groups of patients which are involved and for the economics of the industry. Again, coming to a group of patients who are deficient in a specific protein, the protein alpha-1 antitrypsin, which is very important for the health of the lungs and of the liver. We have a concentrate of alpha-1 antitrypsin, which is, as you can see, widely used now and again has exploded in its use over the past 20 years. Very important for people with alpha-1 antitrypsin deficiency. We have the group of proteins which are dependent on vitamin K, coagulation factors dependent on vitamin K, including factor 2, factor 7, factor 9, and factor 10, which are co-purified in the form of prothrombin complex concentrates. This product is also very important, particularly for treating people who have had an overdosage of anticoagulants for various reasons. And again, another extremely pivotal protein for people deficient in it, C1 esterase inhibitor, which causes hereditary angioedema. This protein is also harvested from plasma. You can see how widely it has increased, and it is a very valuable protein in terms of the sales of the industry. And I would now like to summarize my talk. I have attempted to show you that the plasma products industry has undergone a period of historical development over the past 80 years. It has grown considerably since its inception, but this has happened particularly over the past 20 years as we have discovered better ways of purifying proteins from plasma and more indications in terms of diseases which can be treated. A series of particular proteins have driven this growth, and I emphasize the important role of immunoglobulin. We use evidence-based medicine in terms of the usage of plasma proteins, but we need to do more and to undertake more clinical development and more clinical studies to validate our use of plasma proteins. I refer you to these important documents developed by the WHO in various forms, as well as documents provided by the system in India. These are extremely pivotal documents which are available to you on the public domain. And finally, I wish to thank you all for your attention, to thank the organizers and WHO for inviting me, and to thank the people who have assisted me in generating these slides and presenting to you this talk.